Great. Well, thank you to the NFCR for personal funding for my laboratory, but also for hosting the panel discussion. Clearly, to eradicate cancer or to enable living with cancer, many new approaches are going to be required in addition to immune therapy. And just a little dig to my friend Drew. There's many more cells in the immune system than T cells. And we need to think about them. Long-term joke. So today we're really very pleased to have preeminent leaders in the field that are thinking about how do we translate new observations in biology, chemical biology, computational approaches, and other kinds of therapeutic approaches into new cancer medicines or new strategies for treating individuals with cancer, but also for recognizing how many of these observations can be put together as biomarkers for patient stratification from the front end, but also response and resistance monitoring when patients are on therapy. And so today we have 4 panel discussants. We have questions directed for each of the panelists. And then after introductions individually and some directed questions, we'll open up for questions from you all to each of our participants. So if I could have the next slide, please. Yeah. I'm the token immunologist up here, and it's hard to follow Drew. In fact, I hate following Drew because 20 years ago, Drew told me, Lisa, you know, it all comes down to the T cell. And I argued with him at the time, but, you know, damn it, Drew, you were right. But important to recognize is that tumors are remarkably resilient and have evolved resiliency to evade everything that Mother Nature and biology can throw at them. And so our job is to understand how the tumor is evading the immune system, which is really the strongest sensor in our bodies for any kind of tissue assault. And the immune system wasn't designed to eradicate cancer, it was designed to eradicate bacteria and viruses and other kinds of pathogenic assaults. And depending upon the tissue location, there's a myriad of evasive mechanisms. So if we embrace those mechanisms, it stands to reason then that as tumors evolve, they will evolve also mechanisms to eradicate those evasive mechanisms. And so as we go forward therapeutically, we have to think about targeting the multitude of evasive mechanisms or critical control points in the biology of cancer progression, but also recognize that the tumor is always going to try to win. And we have to be able to have biomarkers that monitor response and resistance as a patient is undergoing therapy and be prepared to sequence those therapies in response to how those patients evolve. In my lab, we use preclinical models to address this in combination with clinical specimens to try to get into the clinic to test hypotheses. And so my bias with regards to asking questions of our panelists will be coming from that point of view. But for them, most of these folks think about tumors from a neoplastic cell intrinsic point of view and how the neoplastic events within tumors drive progression and or response to therapy. So we'll start today with Dr. Bardia. We'll go to Dr. Chernoff, then Dr. Lippman, and Dr. Mischel. And so if you would all please spend 5 or so minutes introducing your points of view and the research in your laboratories, then we'll open it. We'll go to questions. Dr. Bardia.
Thanks so much. Good morning, everyone. It's my pleasure to be here today. It's my first time attending NFCR and I already feel very energized and excited being here. My background has been in drug development, in development of novel therapies. As a background, I'm a medical oncologist. I did my fellowship at Hopkins, and then I was at Mass General Hospital working with Jose Baselga, Daniel Haber and the group, and was involved in development of novel drugs. Over the past 10 years, most of the focus has been on antibody-drug conjugates, developing a drug called sacituzumab govitecan or IMU-132. Then I've been involved with development of other drugs as well. So I'll spend most of my time talking about antibody-drug conjugates and how they are essentially, as I would say, replacing chemotherapy. So the idea with antibody-drug conjugates is fairly simple. You have an antibody that would bind to an antigen that is overexpressed in cancer cells, not so much expressed in normal cells. So you have some selectivity in terms of the antibody. The antibody binds to the cancer cell, then is internalized in the cancer cell and releases the payload, which then causes apoptosis. In the field of breast oncology, the first drug that was approved, which was an antibody-drug conjugate, was TDM1. So building on the success of trastuzumab, trastuzumab usually is combined with chemotherapy. The idea was, rather than giving trastuzumab and chemotherapy separately, can we actually attach chemotherapy to trastuzumab? And that was the drug TDM1. DM1, which is the payload, was developed in the 1970s, and at that time was too toxic to be developed as a chemotherapy agent. But in combination with TDM1, where you're giving more doses of the payload selectively to cancer cell, that did work. Now, TDM1 works very well in HER2 amplified breast cancers. It's approved in that setting. But we also know that tumors are heterogeneous. There can be heterogeneity in the expression of the antigen. And so newer ADCs, that has been somewhat of a game changer, I would say, not just in breast oncology, but the field of oncology in general, is the so-called bystander effect. And the idea is, if the ADC can bind to the cancer cell, release the payload, what about the cells that do not express the antigen? There are two major mechanisms behind bystander effect. The most common one is where the payload is membrane permeable. And an example of that is with trastuzumab deruxtecan. The idea is, again, the ADC binds to the cancer cell, releases payload, deruxtecan, which then causes apoptosis. But deruxtecan is membrane permeable. And because of this property, it can even impact cells in the tumor microenvironment that do not express the antigen. The other mechanism of bystander effect is by the linker. And an example of that is with sacituzumab govitecan, the ADC that targets TROP2. It has a hydrolyzable linker that is pH sensitive. So in the acidic tumor microenvironment, it can release more of the payload, sometimes even before binding of the ADC to the antigen, and thus provide a bystander effect. Moving forward, it brings up a number of interesting questions. The first one, we always talk about biomarkers. So with sacituzumab govitecan, the drug that I was involved with, it targets TROP2. It was first developed in triple-negative breast cancer, and we saw a response rate of 30%, which is much more than standard chemotherapy in that setting. The question was, can we identify biomarkers that can predict who's going to have an excellent response, including complete response versus not? And the first place to start with was TROP2. And it turns out that TROP2 is not a good biomarker for predicting response to sacituzumab govitecan. And I think in part is because of the mechanism of action and the bystander effect. These drugs are very different from the, if you will, first generation of drugs like TDM1. So there was a good correlation between expression of the antigen and response. With these ADCs, the relationship is there, but is not that strong, in part because of bystander effect. Similarly with trastuzumab deruxtecan, HER2 is a predictor, but even lower expression of HER2 is associated with the response to trastuzumab deruxtecan, and that's why we have these so-called new categories of HER2 low, which essentially is a made-up category, does not have biological significance, but the idea is that there is not a very strong correlation between the expression of the antigen and response. The second question then becomes, what are the mechanisms of resistance to these different ADCs? And we can discuss the various mechanisms, but it's quite complex because now you have the antibody, you have the linker, you have the payload, you have processing of the ADCs, and all of these different steps potentially could be involved with resistance. And a third question that tends to come up is, what's the role of the tumor microenvironment and immunotherapy? And as we just heard, with apoptosis, there's activation of the tumor microenvironment. So does the type of payload that's used in the ADC have an impact in terms of how the immune system is activated? Does the type of antibody have an impact in terms of how the immune system is activated? So a lot more needs to be done. This was the first example, at least in breast oncology, where you could deliver more chemotherapy selectively to cancer cells, but the principle is not just with chemotherapy, it could be with other agents as well. Essentially, the idea with an ADC, it's a Trojan horse. You can deliver something more selectively to cancer cells as compared to normal cells, and thus improve the therapeutic window.
Great. Dr. Bardia, so there's over 100 ADCs in the clinic right now in development. Some have made it all the way through into testing. How do we choose? How does a medical oncologist choose which to treat? Are there biomarkers? And then how do we think about sequencing the ADCs with other therapeutics? These would come before a checkpoint inhibitor, an antivascular agent, et cetera. How do we think about that?
Great questions, and something that we are actually grappling in the clinic as medical oncologists. So for example, for a patient with metastatic breast cancer, you have sacituzumab govitecan that's approved, trastuzumab deruxtecan that's approved, and the question is, for a given patient, which one do I start with? As I was saying, the biomarkers in terms of looking at the antigen per se is not a very strong predictor of response. So at least at this time, it's coming down to the side effect profile, discussing with the patient in terms of the schedule and the side effect profile. But hopefully in the future, we can get much better in terms of identifying potential predictive biomarkers of response to these ADCs. It's also an important question in terms of sequencing. If I use trastuzumab deruxtecan first, and a patient then has disease progression, say after 9 months or 12 months, because in metastatic breast cancer, we use drugs sequentially, then after TDXT, can I use sacituzumab govitecan? Both of these drugs have a similar payload. Deruxtecan is a TOP1 inhibitor, and SN38 is a TOP1 inhibitor as well. We did a study looking at mechanisms of resistance, and this is example from just one patient and how we can learn from one patient. There was a patient who had done very well on sacituzumab govitecan, patient with metastatic triple-negative breast cancer, and then about 18 months after she did very well on sacituzumab govitecan, she had disease progression and unfortunately expired. So we did whole exome sequencing of the different lesions that were increasing or had disease progression on scans. And interestingly, in the same patient, we found two different mechanisms of resistance. In some lesions, the tumor had developed mutation in TROP2, so that's the target of the antibody. And in other lesions, in the same patient, there were mutations in TOP1, which is the target of the payload. So it just highlights that there are different mechanisms of resistance, which could be either to binding of the ADC to the target or that mediate resistance to the payload. And this has important implications in terms of sequencing, because if the resistance is predominantly antibody and you use one antibody after other, which is the same, such as TDM1 and trastuzumab deruxtecan, there'll be cross resistance. But if you switch antibody from TDXT to sacituzumab govitecan, that potentially could still work because you've changed the antibody. On the other hand, if the resistance is predominantly payload, then you use TDXT and then sacituzumab govitecan. There's a newer ADC being developed, datopotamab deruxtecan, and there are a number of ADCs with similar payloads that are all TOP1 based. That would result in cross resistance. So it just highlights how understanding resistance potentially could help us with sequencing.
So these aren't frontline agents at this point in time, but in a patient with polyclonal metastases, are you seeing different response rates based upon that? That would be an expectation.
Potentially, it would be an expectation. The other challenge we have is, these analyses between biomarkers and response are in the metastatic setting, and you're doing one biopsy and you're just looking at the expression of the antigen in that biopsy. And we know that there could be a spatial heterogeneity in terms of expression of the antigen. So potentially either functional PET scans, where you get the complete picture of the different antigens, or looking at circulating tumor cells, potentially could be valuable. And that's the grant we received from NFCR, was to look at the expression of these antigens in circulating tumor cells. We cannot look at ctDNA because we are looking at expression of the antigen on cell surface, but CTCs potentially could be a way of looking at the expression of the antigens, and I suspect that would be a better way than just doing a biopsy, at least in the metastatic setting.
Wonderful, thank you so much. Let's move on to Dr. Jonathan Chernoff, who will talk to us about molecular glues.
Thank you. I think I'm the only representative of the East Coast here, so I may be the only person who's actually awake on this panel. But anyway, it's great to be here. So I have two jobs in my career. On the one hand, I'm the director of the Fox Chase Cancer Center, and I guess in some ways is formally my most important job. But to me personally, my most important job is number two, which is that have been and remain an active scientist. I run a lab, I've run one for 30 years. I think we do some pretty interesting things, and that is where my true passion is. I'll talk more about that than my leadership role. So, we heard at the beginning from Dr. Pardal about, by Dr. Cavity about how confusing terminologies are in immunology. That it's a difficult field for the outsider to pierce sometimes. I always felt that way about metabolomics. I look at these metabolic charts and they're just getting more and more complicated until somebody explained to me one day, just chase the electrons. If you do that, metabolism actually makes some kind of sense. And so I have a better feel for the metabolic pathways than I used to. And I think people feel the same way about signal transduction. You have these tremendously complicated charts. There's many important pathways, both cell intrinsic and extrinsic, that seem to govern cancer cell behavior. How do you get that in your grasp? And to me, it's all proximity. Proteins have to be close to one another to do their thing. And if you think about it in that sense, where proteins are, a lot of signaling starts to make sense without having to know every last detail. And we'll just start, at the very top, and we'll hear later about the EGF receptor from Dr. Slayman, I think. There's a thing that doesn't work when it's in a monomer so much. It normally works because a ligand comes along, dimerizes, or a mutation causes dimerization, in the case of HER2, that then sets off a whole signaling cascade. And similarly, inside a cell, proteins generally have to aggregate. Even if we stay with the EGF receptor example, once it dimerizes, then it sets off other proximity agents, proteins that have SH2 domains, etc., that form a conglomerate on the inside of the cell, ultimately leading to RAS and other pathways. But all of them involve protein-protein interaction. And that's the way nature designed it. It's fine, but our ability to perturb that system is where we might make a difference. And now, of course, in a positive way, we've always been able to do it. If you take EGF receptor ligand, you can force dimerization that way, but that's on the surface of the cell. What about all the stuff that's inside? And so there's a lot of pathways that we'd like to interrupt. There are some pathways we'd like to actually promote. Can we do it? And our ability to do it until recently has been pretty limited. But there's always been these couple of peculiar molecules around, which we now call glues, but back in the day, people didn't really know what they were. I mean, things like thalidomide, which of course has a long and somewhat sordid history, but then came back as an agent that's useful in itself, but also as a platform for other type of drug development, which end up forcing proteins into proximity that either should be in proximity normally or caused to come into proximity by these novel small molecules. And so there's a number of these out there. that nature's designed, but it's occurred to people that you might be able to design them on your own to therapeutic benefit. And so there, you get into this whole issues of things like molecular glues and PROTACs, which have become very popular lately. I show one example on the slide behind me of one that's made a huge impact recently in the RAS signaling field, which is, of course, was an undruggable target 10 years ago, eventually became druggable, but now with this new concept of using a glue to basically stick cyclophilin on top of RAS and prevent it from interacting has really revolutionized how we look at things like pancreatic cancer. In fact, the company that makes this one is called Revolutions Medicine, and by the way, I do not hold stock in them. I'm not giving them some free advertising here, but a lot of people have now converged on this concept of forcing proteins into interactions which may be productive for us in terms of blocking proliferative signaling. So then the issue is if you can do this sort of thing, it's a pretty neat trick, and how do you design them? And that's been a kind of a roadblock to date. Most of the molecular glues that are around have been accidentally discovered. Thalidomide being one example, or the compound, I don't know its name, that Novartis makes, that's a SHP2 inhibitor. That was designed by conventional means as an inhibitor, and only later in the analysis did they find that it's actually acting as a glue, kind of holding this protein in an inactive conformation. So how could you do it de novo? And here comes a new other technology, which is one I don't understand very well, which is artificial intelligence and machine learning. So in modern drug design, or in today's drug design, it's not like it used to be. It's changing almost by the week now. Enabled by programs like AlphaFold and other similar computational tools, these AI chemists can now survey not just millions, billions, and even up to maybe 1 trillion different elements of chemical space and come up with predictive glues. And so you ask, I would like to glue protein X to Y. We'll just leave the identities of X and Y blank for the moment, but it could be almost anything. They will survey chemical space for a potential glue that might hold these two things together. You then are presented with a small library of maybe 10 or 12 compounds that have been predicted, and you can get those made, and then you assay those rather than starting with 100,000 compounds and trying to do assays with all of them, which is what we did a few years ago. I think we're on a cusp of some pretty amazing tricks in small molecule biology. I know we've heard a lot about immunotherapies and so on, and I am totally in favor of that approach. In fact, I think all of these are going to end up being combined, nature being as clever as it is in evading what we do. But this approach is really going to present some new opportunities for drugging things that we have not been able to see before. Now, to quote that line from Jurassic Park, I think, life always finds a way. You mentioned this in your opening comments. We make these glues, including the one shown here for RAS. Tumors do find a way around it eventually, and we need to do a combined approach. That's going to be pretty clear. There's no true magic bullet out there that I'm aware of. But this development in glues, I think, is very promising. And even my own lab has now gotten into this in a heavy way, which maybe we'll talk about it at a future meeting. But I think it's a very promising development.
Yeah, thank you. So how do we think about these in the context of the natural resistance that emerges after targeted therapy in patients?
I think most people in the audience understand that we help provide our own questions beforehand. And of course, when I started asking some of these questions, it's because I don't know the answer. And so I'm hoping that my very clever co-panelists or audience will help me here. So, resistance, this has been the bugbear of all of our approaches to date. I mean, we go all the way back to the original therapies for bacterial infections and, quickly emerge resistant clones. Well, the same thing happens in tumor cells. We have excellent designs, great targets that we validated, and then 2 weeks later, resistant clones emerge. Often enabled by the surrounding cells, but often also intrinsic to the cancer cell. I think, we should just understand that that's what's going to happen. Don't be surprised every time you have a great agent and you find resistant clones. That is kind of the going expectation. So, there are some conventional ways to analyze and try to defeat this. Synthetic lethal screens are the most common one now, using either CRISPR, RNAi, or drug screens. Those are pretty good in that you'll find a secondary target that emerges, but oftentimes you then go after that one and yet a third thing comes up. I think at some point you will checkmate these tumors. It may take even a triple approach. I'm not sure if that's going to be true for all of them, but I think if we hit different arms, different vulnerabilities at the same time, it's going to be very difficult for resistant clones to emerge. So that's my hope. It's not that there's going to be a super drug, one thing. It's going to be a couple of super approaches combined together, almost certainly involving the immune system as one of the arms of that.
So you mentioned RAS and our ability now to drug what we thought was once undruggable. The natural biology of RAS we now appreciate is, in addition to intrinsic signaling, is a master regulator of local immune response. So how do we now integrate the glues for RAS in the context of the diversity of immune therapies that we're also putting into patients?
Yeah, no, a great and another question that I can't really answer definitively, but I can see how it might get answered. So I think, our understanding of what RAS does to the extracellular environment is imperfect right now. I think within the cell, we have a pretty good grasp, and it's been studied now for 40 years. Not that there's no surprises left, but I think we know most of what RAS does, which basically is accurate ERK, in my opinion. But outside the cell, it's a little different. There's different effects on different pathways. We saw the complexity of the immune environment in some of these single cell sequencing efforts, and I think we need to get a better handle on those to predict what's going to happen when you start impeding this pathway. And one technical plug that I'll put in here, because we haven't seen it yet today, certainly the single cell analysis has been revolutionary for everything we do. What hasn't come quite yet to bear, but will in a couple of years, is single cell proteomics. To me, as a signaling guy, it's not that I don't want to say I could care less about RNA, because that would be overstating. But really, the correlation between RNA transcript levels and protein expression is not perfect by any means. Some studies have it, around 50%. So I'm really interested in what's going on at the protein level, what's phosphorylated, what signaling pathways are actually on. We've been stymied in our ability to do kind of single cell equivalent analysis of that. I mean, you can do it with antibodies to some extent, but that's just limited to your antibody pool. But having talked to some of the proteomics experts lately, I think in a couple of years we're going to be doing the same thing that we've seen with RNA-seq and DNA-seq with proteins. And the reason I mention all this, I think that will help enormously in understanding what's going on in the extracellular environment as well. What the signaling is in the T cells and the NK cells and all of the surrounding fibroblasts, etc. We don't know that yet, but I think we will soon.
So that company will be called 100X.
Right.
Because our NIH grants certainly will not provide funding for that. Thank you so much. We'll move on to Dr. Lippman from UC San Diego.
Thank you. I initially thought we had 2 minutes, so I'll just talk slower. Which is a normal speed for most people coming from New York. But yeah, no, I also have a Hopkins connection from medical school and an internship actually a year or two behind Drew, who's much older. But
I was at Hopkins too.
I'll be here all day if people want stories of that time, Drew stories. Yeah, I was at MD Anderson for 24 years, department chair, very large department of medicine, and then I moved to UCSD about 14 years ago. Yeah, I mean, Drew actually mentioned that having a strong collaboration with a computational biologist is important, and that's really what happened to me. I'm involved in translational research, but about 5 years or so ago, I was very fortunate to recruit a very deep collaboration with Vladimir Alexandrov, who discovered mutational signatures and clusters, and now some of the AI work that I'll show. Also, a wonderful collaboration with Teresa DeVoli, who is here, who's also a brilliant computational person and discovered association with copy number alterations with immunity and immune checkpoint resistance, and is doing work, NFCR funded work with Sylvia Beckman, who's also here. So the work that I'll talk about is some fairly recent, although we've been working on it for a few years, work involving artificial intelligence in oncology, specifically deep learning AI. And just a little bit of context about this, the field of precision oncology really began with HER2 and really the breakthrough work of Dennis Slayman and others about 25 years ago. And it's continuing to grow. In fact, recent estimates over the past 5 years are that the rates of actionable events have increased about fourfold. So there's a lot of work going on, there's a lot of reasons for that. But at the same time, very large databases, ASCO databases, and others have actually done some very sobering work looking at actual rates of genomic testing. And again, precision oncology, by definition, requires some sort of molecular profile. And what people have found, and this is the United States, is about 22 to 25% of patients with the poster child for precision therapy, lung adenocarcinoma, are actually getting FDA-approved level 1 companion diagnostic testing. This is in the package insert required. It's about 25%. And in work that was done by Mischel and others, published late 2022 in New England Journal of Medicine, sort of a call to arms on this idea. The rates of genomic testing, FDA genomic testing, was in the US about 22% in whites and 11% in black patients with lung cancer. And there's a lot of work looking at low socioeconomic status. Rates are lower. Global testing rates of standard tests, the standard NGS like Foundation Medicine or Tempus or so on, is 4%. So this has really shaken the field up, because you start asking, what's the point of doing these great trials and all this work if patients aren't getting tested? This is a real crisis. And AI is involved in virtually every aspect of oncology, and arguably the most important focus right now is what this work is addressing, and others as well. And that is there a way to facilitate, expedite, increase the uptake of standard genomic testing? And the problems, the reasons that genomic testing rates are so low is about 4 major reasons. The time, I'll come back to, the cost of testing, the tissue requirements of testing, and the complexity of the whole process. I don't know how many people clinically, are involved in this testing, but if you've ever seen I will name it, company report on one of your patients, they're like 26 pages long, with all of these variants of unknown significance, this and that. It's very challenging. So even the young oncologists coming out, academics, are starting to pull back on testing, because they're forced in a situation where you order the test, and a test itself can take 3 to 6 weeks. And there's usually a 20 to 30% sort of QNS, not analyzable result. Then you're sitting like, what do you do now? Do you biopsy, get more tissue? The whole process is really never been baked in fully to oncology workups. And so, because of all those problems, people look in different ways. And so the way that we started looking at was to sort of ask the question, in routine H&E slides, which the H&E stain using a light microscope were invented over a century ago. We're going to Galileo and things like that. And the scopes are a little better now, but they're really the same. And they look like that upper left-hand image. That's a routine biopsy with an H&E stain. It's done everywhere in the world. It's done everywhere in the world. I mean, a Nigerian farm town have the ability to cut a slice and do that stain, the routine stain. So that's available. And it turns out that, and again, I didn't know any of this before, but it turns out that an H&E slide is incredibly data rich. It's like 15 to 50 gigabytes, over 100-fold larger in terms of the data size as like a CT scan, for instance. And so that's a double-edged sword, but the positive is it might be possible to pick up subclinical patterns of cells within an H&E slide, like a microscope, that a great pathologist or a normal eye would not be able to detect. And that really is what started this whole area. And And so, the answer is it looks like it can. And before I show the results, what that would do, we're talking about extracting actionable genomic events. Now, we can talk about what the definition of actionable is. It's like everything else. There are levels of actionability. But in any case, of companion diagnostic actionable events, can extract actionable information from a standard HCD slide. This requires the same actual diagnostic slide at the same time the pathologist is doing this. So pathologists, when they view slides, they do multi-resolution. They go low resolution, 5x, then go to 20x. I don't know what 20x would fit. We've seen 10x and 100x mentioned, but high to low resolution. And the deep learning model can do that a million times back and forth. And so anyway, it's pattern. That's what's shown at the top. So you're using the same diagnostic slide. You don't need any more tissue. You don't even have to cut another section. It's very inexpensive because you're not sequencing, which costs about standard tests are $3,000, give or take. And the way this was developed is with clear thresholds. So there are no borderlines, there are no QNS. Every slide, and there are thousands that London scripts looked at, every slide that could be read by a pathologist could be read out. In the case here, which is a genomic event of homologous recombination deficiency, but there are a number of other genomic markers as well, as a positive or negative. There's no, and in some fields, it's very frustrating to get these borderline results, even flipping a coin. So it has a lot of advantages. Anyway, and so the way that this was developed is a little different. This was trained, if you look in the upper right, you see the AUC curves. It was trained on fresh frozen and paraffin embedded slides, over 1,000 each, and the AUCs for detection of just genomic event were 0.81 in both cases, different 95% confidence intervals. So first shown that it could detect the genomic event with good accuracy, and independently tested to see if it could predict outcome for drug targeting that genomic event. And remarkably, it performed extremely well. It's perfect. Progression-free survival curves in the upper right in breast cancer, with the red is the sort of HRD predicted, and blue is the HRP resistance. And it actually was compared head-to-head to, you've heard about ground truth from Drew, the FDA, in this case, the FDA-approved Myriad test, as well as the SBS3 mutational signature, BRCA1/2, and then a 26-gene panel of Palbumine and others, and outperformed all those, not only in terms of predicting outcome, but in this case, actually capturing more patients that could benefit. And then as you see the bottom that just shows the various aspects, I think in an earlier talk was mentioned about the CNN network and MIL learning and things like that I don't actually understand. Douglas tried to explain it to me several times, but I've seen the data. So I think I'll stop there. If you can maybe click it, you'll see the overlay of sort of the pixels, where on the bottom right you see the H&E slide. This is a triple-negative breast cancer patient. And then the pixel overlay shows the location of the HRP and HRD areas. The blue is HRP and the red is HRD. So you can kind of see these patterns, and then you can start getting into the black box issue of what is it that this AI platform is using, it's true of any platform, to make its prediction. And that's what these pixelated overlays show, and we can talk about what that was. The editorial in this paper from JCO came out last month and referred to this as the next wave in precision oncology, the first wave being starting with HER2, because this is literally a crisis in precision oncology. If we can't get testing rates up, it really raises the question, where to go from here?
So given the heterogeneity in any one tumor, how do you anticipate efficacy here with prediction in a patient with polyclonal disease? If this is AI on a single H&E, how do we embrace heterogeneity to treat the patient?
Yeah, you can see the heterogeneity spatially on the bottom right of that slide, showing the different areas. I don't know how the platform works, how Leadmill takes that to then determine whether it's HRD overall. So I won't even try to guess, but at least, figuratively you can see that it is capturing this heterogeneity, and you can see the areas that are HRD positive in this case. But how all of that comes together to then end up with a final prediction, it involves like thousands of iterations and models and things that I really don't understand. I apologize.
And do you see this as applicable across the community?
Oh, definitely. Yeah, I mean, there's work to be done, but there's no question. I mean, I see patients, there's no question this is the future. I don't know if it'll be this platform or some others, but this approach is the future of precision oncology, no question in my mind. Now, it's not going to replace liquid biopsies and other sort of things, but for really expediting first-line therapy. And just to be clear, the time from diagnosis, from your H&E diagnosis to starting therapy is the major determinant of patient outcome and survival. The major determinant. In fact, there's a paper that came out about a year ago that actually looked at that rate. And in lung cancer, again, the poster child, for every week from the H&E diagnosis, the pathological diagnosis, to starting therapy, 4% of patients die. 5 weeks, 20%, and many more of those patients become too ill or frail to get therapy. So, and you can't even do that analysis in pancreatic cancer because you can't wait. This test, this HRD, happens to be a very good test in pancreatic cancer for platinum and PARP inhibitors, and it's recommended to be used, but it's never been tested prospectively because it would be unethical to delay the treatment of pancreatic cancer a few weeks when, as we all know, pancreatic cancer survival is measured in the order of months.
Great. All right, thank you. We'll turn now to our last panelist, Dr. Paul Mischel from Stanford University.
We thought we understood cancer genomes. We had a map, right? We knew where all of the genes were on chromosomes. And we had next generation sequencing technologies. So we could take a tumor from a patient, we could grind it up, we could extract the DNA, we could see what had gone wrong, and we could develop precision oncology for those patients. And in some cases, it worked. But there were a lot of patients for whom it didn't work at all. And the problem is that something was missing from the map, and that's extra-chromosomal DNA. You see in the picture, that's an electron micrograph of the DNA inside the nucleus of a cancer cell. And you see the chromosomes, the beautiful shaped bodies there. And then you see those circles. Those have been hiding in plain sight inside the nuclei of cancer cells at a remarkably high frequency, wreaking havoc. And here's the problem. When these oncogenes untether themselves from chromosomes, they break Mendel's laws, and they start becoming inherited in a completely different fashion. I'm going to give you an overview of work that's happened over the last 10 years. And I'm going to start with the nature of the problem and what got us there. I was really troubled by a few things. One, how is it that tumors are so heterogeneous? We really didn't have answers to that. Second of all, how do you get 40, 50, 60 copies of an oncogene in a cancer? I went back to the original literature of amplification, and I saw a bunch of reviews that cited a bunch of reviews with almost no data that explained how this was actually happening. And with the exception of HER2 and breast cancer, patients whose cancers have oncogene amplification do much worse than all other cancer patients. And nobody actually understood why. So we ended up doing something that sounds remarkably simple, but hadn't actually been done, which is instead of just reading sequences, we actually looked. We looked at location and we looked at shape. And what we found was that, in fact, many cancers had extra oncogenes that had untethered themselves from chromosomes on extra-chromosomal DNA. And when you treated with targeted inhibitors, the DNA seemed to disappear. And when you removed the drug, it reappeared, and it all happened very quickly. Cancers were changing their genomes fast, and that made no sense by classical genetics. But you see, when they jump onto these circles, they don't follow Mendel's rules. So let's think about why that matters. Plants and animals are multicellular. Chromosomes evolved so that during a cell division, each cell gets the same DNA. The daughter cells have the same DNA, and therefore you can build a body. Let's say you had 3 copies of genes on chromosomes. When the cell is getting ready to divide, it would make 6, and it would give each daughter cell 3 copies. But if it were on extra-chromosomal DNA elements that lack centromeres, those 3 would become 6, but they could be parcels 6 and 0, or 5 and 1, or 4 and 2, or 3 and 3. Notice 2 things have happened. You've doubled the potential ceiling. Play it forward now. And now is how you get 40, 50, 60, 70 copies. And the other thing you've done is you've increased variance. And Darwin taught us that's the fuel for evolution. Every cell division with extra-chromosomal DNA is like a dice roll. You get a random bell-shaped curve distribution. That challenges our very notions of clonality and explains why cancers can evolve so quickly. After publishing the first paper in Science in 2014, I began to realize there was a problem when I would go and talk to people. We were studying EGF receptor at the time. And I'd give talks, and people would say to me, you know, that's interesting. The oncogene I study, Nick, it's on chromosome 8. And I would say, how do you know? And they'd pull out a TCGA map, and all I thought to myself was if Ptolemy's map of the solar system, maybe the measurements were right, but the map was wrong because of the orientation. So we began to look at this. And I'm going to tell you about work that happened over a span of 10 years. First, with a small group of us, and then over the last 3 years, through the Cancer Grand Challenges program, which has allowed us to work together in a very unusual way. I have the privilege of leading a team with extraordinary scientists, people like Howard Chang, Vineet Bafna, Ben Cravat, James Chen, Serena Nixino, Miriam Jamal Anjani, Jeff Buka, Harmit Malik, amongst others. It's an unusual group of people that work together in a completely different sort of way. Here are some of the things that we've actually learned. The first thing we've learned is that this mechanism of inheritance is completely different. That is, random segregation is very powerful, irregardless of the oncogene and irregardless of the tumor type. This is how cancers maintain this heterogeneity, because each cell division is a dice roll. Further, when you treat, because you have a bell-shaped distribution, they can change their genomes within 2 cell cycles. And that's very powerful. We've also recently learned that it changes how genes are transcribed, because it's not just a genetic problem, it's an epigenetic problem as well. The circular DNA is chromatinized and wound around histones with nucleosomes and cores, but it's highly accessible. It doesn't hypercompact. It's loaded with active histones. It has a paucity of repressive histone marks. It transcribes over 4-fold more than chromosomal DNA, even when normalized for copy number. It changes how genes are regulated in cis, as we know how genes inside enhancers talk to promoters inside loops. And now you've got these loops on a string, and you've got insulators between the loops, and things that are far apart don't talk to each other. Take the ends of the string and put them together into a loop, and you can see 2 things that have happened. One, now everything is close, and the insulators no longer matter. It's not only cis regulation that's changed, it's regulation in trans, because my colleague Howard Chang, and we worked with him on this, they come together in hubs where 2 circles can interact with each other, an enhancer on one talking to a promoter on the other. And that's a very powerful mechanism. We've then gone on to show that, in fact, this doesn't just happen late in cancer. It can happen early. When we looked at patients with Barrett's esophagus, they can get ecDNA after losing P53 during high-grade dysplasia. And when they get it, they will go on to get cancer, which opens the door for the whole idea that we need to be thinking very hard about early detection. We now know that ecDNA is a huge problem. In a new collaboration we've done, my team with Charlie Swan's team and TracerX, in 15,000 patients in genomic England, the scale of this problem is huge. 17.1% of all cancers, and that's biased towards early stage. Much higher levels in advanced cancer and metastasis. And patients whose cancers have extra-chromosomal DNA do worse than all other cancer patients by far. And it's not only oncogenes that they contain. They also contain regulatory elements. It turns out immunosuppressive elements as well. These tumors are stone cold for those reasons. We've also now gotten to a point where we've begun to understand the molecular mechanisms that are underlying it and have the first targeted therapies that are now being tested in phase 1 clinical trials in the clinic. I know I'm out of time, and I've put forward something that's very different, but I'd like to point out to you that extra-chromosomal DNA is a huge challenge. In the spirit of Dr. Albert Giangiorgi, who said that research is about seeing something that's been seen before but not understood, ecDNA falls into that category. It was first seen 60 years ago, but not understood. And when I started working on it, it was thought to be 1.4% of all cancers and of unclear significance. Now, the mechanism is very clear and the importance is very clear. One thing that I hope will come out of this conference and this meeting was that the community will galvanize to realize the scale of the problem, because these are the patients who are being left behind.
Great, thank you. So one quick question for you before we turn it over to the audience for questions for the panelists. How do we turn these into actionable therapies?
Yeah. So what's very interesting is that, and we're working very hard on this, and I hope the large community at large will work on this. There are conventional targets that are likely to be shared by many sorts of cancers that have oncogene amplification, in which there's a particular dependence in ecDNA. The first one has just been identified. We've identified that because of the open chromatin, there's replication transcription collisions that are occurring at massive scale that makes them extraordinarily vulnerable to CHEK1 inhibition. And the first drug produced by Boundless Bios now, and in full disclosure, I'm a founder of that company, is in phase 1 clinical trials. But we think this is just the tip of the iceberg. Quite likely, many of the proteins that are going to be involved in ecDNA biology that are novel are likely to fall into traditional undruggable classes. So what you mentioned, Jonathan, is certainly of interest to us. And we're working very closely with Ben Cravath and his unique chemistry to develop ways with cysteine liganding to go after these unique targets. So I think we're at the beginning of something.
Great, okay. Now, we have roving microphones for anyone that would like to ask the panelists a question, but please wait. Dr. Weaver.
Yeah, so this question is for Paul. I'm very intrigued by, this extra chromosomal DNA and these oncogenes. Do you know what actually initiates the generation of these? And is there evidence that specific oncogenic mutations in the cells make them more susceptible to actually having this occurrence?
Great questions. We actually do know. So let me start with the second part. What is the genetic background in which it occurs? P53 loss is a huge player, but it's tissue context dependent. For example, in some sarcomas, it's an NF1 loss. Those data are now, will be out there very soon. P53, though, turns out to be a huge player. We think suppressor losses play a critical role. We also think there may be underlying defects in the DDR system that we don't yet understand that might be involved in this, and we're looking actively for that. Now let's go to the second part. How does this actually happen? So this is what's known. One mechanism is through chromothripsis, and this is work from people like David Pellman and Don Cleveland and Peter Campbell. When a chromosome lags, it can go into a micronucleus and get chopped to bits. Now it's interesting, my lab was close to Don Cleveland's lab, and I don't think Don believed our work on extra-chromosomal DNA until he made a chromosome that lagged. Peter Lee did this, and the only time it amplified was when the pieces reassembled into a circle. And then Don became very focused on this, as is David Pellman. So chromothripsis is very important, and it's one mechanism. We think it's about a third. The other thing we know is that paired double-strand breaks in the context of a P53 loss or another suppressor loss can do it. Now that raises a really interesting thing, because you're only going to see what is selected. So we know that this is a process that continues, but if it and the genome is a very large space, so it's not like there's a particular region that's vulnerable to it particularly. Rather, the landscape screens selection. That is, if you pick up a MYC here, or you pick an EGF receptor here, or you pick up a cyclin D1 here, and it's driven actually by tissue context. So we know those are at least two mechanisms by which this occurs. There may be more.
Great, thank you. Question here?
I have a question for Scott. I grant you that little biopsy can inform you quite a lot about the tumor, but there's still a little piece of tissue out of a big tumor, not representing potential metastatic sites. What about integrating that with liquid biopsy? All the microRNAs, the exosomes, the proteins, the metabolites, and everything else being released you would get a much better picture of the whole tumor than just looking at that little biopsy. Wouldn't you agree?
Absolutely. I mean, this technology doesn't replace liquid biopsies. It doesn't replace NGS. It really complements it. Really, in this very specific situation of clear, actionable events that are established and getting patients on therapy quickly, where you don't have to wait for and just sequencing and so on. But clearly, those others will help fine tune, and they all sort of work together. The heterogeneity I meant to mention this is that those groups of pixels that are blue or red have been termed superpixels, and somehow they get calculated or weighted differently in the ultimate model prediction. But Sam, I definitely agree with you. This is just one technology that really will help complement the others.
Thank you.
So I won't build on Dr. Pardo's last sentiment in terms of IO combining with other modalities, and being a chemo developer and IO developer, I've never seen anything so effective than the PD-1 combined with chemotherapy, both from indication and also therapeutic impact to patients. So this is actually a question for Aditya. So now seeing ADC as the next wave of more effective chemotherapy, if you see envisioning this going to converge with the IO therapy or the other modalities, and how can they be combined?
Absolutely great question. I think there are two components to this question. The first is looking at ADCs as a way of delivering more chemotherapy to cancer cells and combining that with immunotherapy, and we have seen that work. So an example I'll give is with the TROP2 ADC plus immunotherapy for patients with triple-negative breast cancer. If we look at the PCR rate or complete pathological response rate with single-agent ADC, a TROP2 ADC, it's about 30%. But if we combine that TROP2 ADC with immunotherapy, as was done in the I-SPY study, the response rate is about 55%. So that's almost double the response with the same ADC, but combining that with immunotherapy. I think the second component we have to consider is also the antibody. That might also have an impact. So for example, if we look at TROP2, TROP2 is involved in cell-cell adhesion, and that can potentially act as a barrier for the immune system to come to the tumor microenvironment and the cancer cells. And if you look at the correlation between TROP2 expression and TILs, you do see a correlation that tumors that have high expression of TROP2 have lower TILs and vice versa. But if you are using an antibody against TROP2, in a way you're allowing the T cells or the infiltrating the TILs to enter the tumor microenvironment. So that would be the other component that we have to look at, is just not the payload, but also the type of antibody that's used when we're combining with immunotherapy.
All right, do we have any other questions for the panelists? All right, well, we're right on time, Sejong, so you'll be very happy about that. If you'll join me, please, in thanking the panelists.